Okay, good afternoon, everybody, and welcome back. So today we're going to begin our discussion about metabolism. This will extend over several classes. And today we're just going to be discussing an introduction, and it's largely a consideration of how cells deal with energy and with matter. So the very first thing to remember is that we gave that as one of the definitions of life itself. That life was something to do with the way in which energy and matter were managed inside a living cell. And that is our consideration today, because our cell is really like a small factory where many complex reactions are occurring. It's important to realize that the cell is actually an open system. That is, material comes into it, material passes out of it. Energy, it comes into the cell and is also lost from the cell, excuse me, as we will learn in a little while. And we fit into a biosphere, which has its own characteristics. First of all, our biosphere is open to energy. We receive energy from the sun largely in the form of light, which is converted into useful energy by plants and then to the rest of the biosphere. But matter in the biosphere must be recycled. Whereas open for the cell, it is closed system for the planet as a whole. Now, we also need to realize that our discussion is very largely going to be about heterotrophs, such as we are. And in heterotrophs, the cell, it will extract energy from fuels of some sort, will actually convert it, and will apply that energy in a biologically useful form to do work within the cell. And that is going to be our consideration today. How is energy dealt with, and how does it fit in with the management of matter within the cell? So you will hear this word metabolism used often in common parlance. It's used quite loosely. And when you talk about metabolism, you sort of think about, well, it's something to do with the cell being alive. Well, metabolism is actually those processes which transform matter and energy within the cell. And it's the sum total of all of the cell's different reactions. We recognize two parts to the metabolism. The first are the catabolic reactions, or catabolism. Catabolic reactions break down molecules. There are various reasons that needs to be done. The principal one that we will be looking at are the catabolic reactions, which extract energy. And that energy is going to be being stored in some molecule. And that molecule needs to be broken so that the energy can be extracted from the bonds in the substance. So that is catabolism. It is usually results in the release of energy, which can then be used. Anabolic reactions are the reactions which build molecules. They build the biomolecules, and they usually require an input of energy in a biologically useful form. Notice that I'm being very careful how I talk about energy. I'm emphasizing all the time that inside the cell, we need to be using energy in a biologically useful form. We require energy in a biologically useful form. We can't use all forms of energy. Light, for example, to us as heterotrophs is useless. Heat is useless. It cannot be metabolically used. So we need to think a little bit about energy itself. Energy is the capacity to cause change or to do work. If you want to change something or you want to do work, you require energy. And as I've said before, energy exists in these various forms, but only some of them can perform work, especially inside the cell. And life requires biologically useful energy. And I'll just mention, we won't talk about this much now, but we will hear much more about it later on when we talk about specific reactions. Inside the cell, the energy that does not simply fly around like lightning bolts. Energy is actually contained in the electrons. Electrons can contain energy, they can absorb energy, they can be excited to high energy states. And we actually, what we're always doing is we're actually shuttling electrons along with their attendant energy around. That's how the cell does it. And we refer to that as reducing power. If I add electrons to something, I reduce that substance. So the cell always requires, in order to perform all the reactions that we're going to, metabolic reactions that we're going to talk about later, the cell actually requires a source of electrons. It requires reducing power. And the energy in the cell is actually locked into those electrons. That's how we shuttle it around. We may shuttle it in specific substances, but remember always that it's actually the electrons associated with those substances that contain the energy that we want to access and use. So there are various kinds of energy we need to think about. The first one is kinetic energy. And kinetic energy is the energy associated with motion. All molecules, for example, have a kinetic energy. As long as they're above the temperature of absolute zero, they are always in motion. It's just random motion, and is simply associated with the nature of being an atom. Kinetic energy is any energy associated with motion, all the way from the atomic level, all the way up to me moving my arm. Heat is thermal energy, and it is also kinetic energy. It's kinetic energy because the heat is associated with random movement. And the higher the kinetic energy, the faster atoms will move. Potential energy is latent energy. It is energy which is kept in a latent form because of its location or its structure. What is really important in biological systems, we often build up potential energy in chemical bonds, because chemical energy is actually potential energy. Chemical energy is the energy associated with bonds, and is in fact also potential energy, which the cell can access. Now, here's the very first thing that we need to remember about energy forms. The energy content of the universe is a constant. Energy can be converted from one form to another, but it cannot be created and it cannot be destroyed. That is also known as the law of conservation of energy. But it's these energy conversions that are so interesting in terms of the metabolism. Now, here's a simple example, not of metabolism, but of how energy forms can be transformed from one to the other. People swimming here are living organisms, right? And they have all sorts of metabolic processes going on. And those are chemical processes. Those are chemical processes which are driving what they're doing. Their swimming is kinetic energy. The energy is derived from catabolic reactions. It is converted into motion of muscle fibers and everything into movement, and that itself is kinetic energy. Somebody lying in the water, then climbs out. When they ascend, they're moving against the force of gravity. And they're having to put energy in to move against the force of gravity. They ascend the diving board and they have acquired more potential energy than somebody down here. If that person then dives off the diving board, that potential energy, which by the way originated from conversion of fuel and of chemical energy in the bonds of the fuel that they're burning as they climb, that potential energy now is converted into kinetic energy, energy of movement, because they are moving, they're falling in the direction of gravity until they hit the water, of course. And once they hit the water, they have less potential energy than the person standing at the top. Energy here is converted from one form to another. All the way along the line, we have to realize, though, that all of these conversions are inefficient. And that is, the first law says energy can't be transferred or transformed. Energy can be transferred and transformed, but it cannot be created, nor can it be destroyed. It's called the law of conservation of energy. But the second law is the killer for biological systems, because every energy transfer or transformation, some energy is lost as heat. Every energy transfer or transformation is inherently inefficient. And that is because of the second law of thermodynamics, which we won't go into in huge detail, but it says every energy transformation must contribute to the entropy, to the disorder of the universe. Some of the energy has to contribute to disorder. And the way in which that happens is we visualize it as each transformation is inefficient, and some of the energy is lost as heat. If you want an example of that happening, put your hand on your arm and feel the warmth coming from your own cells. That warmth, although it's useful to us because we keep a constant body temperature, we can't use the heat. There's no way for biological systems to fix heat energy and transform it. It's the lowest form of energy, it's the lowest grade of energy. And it's not, in fact, useful to us. It is waste energy. It radiates from your skin because your cells are performing all of the metabolic reactions that contribute to life. A certain amount of those energy transformations that are inefficient, and they release some energy as heat. That heat is eventually radiated out to space from the planet. So that is why I said at the beginning, our planet is open for energy. Our cells are open to both these and matter. The planet is open to energy because energy coming in as light undergoes all of these biological transformations, but eventually is radiated back out to space as heat. Because of the second law of thermodynamics, every transfer or transformation of energy is inherently inefficient. So here we are. Here's our, this is appropriate because this is exactly what bears are busy doing at the moment. They're busy building up fuel stocks to see them through their hibernation during winter. So here's the first law in action. This bear is eating fish, and it is a heterotroph. The bear is a heterotroph. So what it is doing is it's taking all of those complex molecules contained in the fish, and it is transforming them. So it's transforming all of the molecules that make up the fish, and it's using both the matter contained in the fish to build up its own body mass, but very importantly, it's using the energy. The energy is potential energy locked into the chemical bonds that make up the fish's macromolecules. So the bear is using fish as both a source of matter, biomass, but also as its source of energy. How it does that, we're going to find out much later on when we talk about how we actually use fuels to drive our metabolic reactions. The second law of thermodynamics says that during all of those transformations, each transformation is inefficient. So, for example, here's the bear running through the water. It is converting fuel energy from the fish into kinetic energy. It's transforming the chemical bonds, the potential energy locked into the chemical bonds. It is transforming that energy into actual kinetic energy. That is necessarily inefficient. And part of in breaking those molecules down to simple compounds, carbon dioxide and water. It is extracting energy to drive the muscles to produce kinetic energy, but it is inefficient, and it is releasing some of that energy as heat, which is then lost to the biological systems. So these questions of energy are actually, at base, the rules sound simple, but in actual fact, they are really complex. Now, in your text, there is a lengthy discussion about the role of Gibbs free energy in driving reactions. I am not going to go over that. Okay. If you wish to do so, you're more than welcome to read all of that. And I'll also tell you that if your understanding is not clear, then there are numerous YouTube videos, for example, in chemistry classes and things, which will discuss it, but I'm not going to discuss it here, except in the simplest terms. And the simplest way to express this is to say we're going to look at two kinds of reaction. The first is called an exergonic reaction. And an exergonic reaction results in the release of free energy. It proceeds with the release of free energy, and it is the reaction proceeds spontaneously to an equilibrium state. When you look at the free energy, which is expressed as delta G, you'll see that in these reactions, the delta G is negative. And that is why the reaction proceeds spontaneously. An endergonic reaction is one where we have to contribute free energy to the reaction. We actually have to put in energy, and the reaction is non-spontaneous. There we say that there is a positive delta G. Exergonic reactions are reactions which are typical of catabolic situations. For example, if we have a fuel or something like that, and we burn that fuel in any way, we burn it physically, or if we burn it in the cell, then that is an exergonic reaction. It's going to release energy. Okay. An endergonic reaction, those are the reactions of anabolism. Anabolic reactions are endergonic reactions. They absorb free energy, and they are not spontaneous. Sorry, I'm scaring my cat away. So exergonic reactions proceed with net release of free energy, and they're spontaneous. Endergonic reactions, they require us to put in energy, and they are not spontaneous. So have a look here with just a simple diagrammatic representation. If we were thinking about the free energy, here it is on this axis here, and we have a set of reactants. In an exergonic reaction, that reaction proceeds to produce products, and in the course of the reaction, it releases energy. If we were to measure the amount of free energy, we'd see that the amount of energy released is less than zero. Delta G, the amount of energy released when we calculate the delta G is less than zero. In an endergonic reaction, we have reactants which we need to put together, but they will not spontaneously they react with one another. Instead, we need to put in energy in order to forge those bonds to make the products. This is very typical of anabolic reactions in biology. We have reactants which will, under normal circumstances, do not react with one another with any rapidity at all. Instead, we have to put in energy to forge those chemical bonds. We end up with products. Here's the amount of energy that is required. And if we calculated the delta G, we'd find it was greater than zero. So exergonic, delta G is less than zero. Endergonic, delta G is greater than zero. But just think about this for a minute. How about, here is a reaction where I require this amount of energy. How about I couple that reaction to this reaction, which would provide me with that amount of energy? Remembering that some of the energy will have to be lost as heat. Okay. But as long as this reaction provides more than enough energy for this, we can couple these and have the whole set proceed spontaneously. And that is exactly what we're going to see happens in biological systems. We couple exergonic reactions to endergonic reactions in order to fund the endergonic reaction. Now, there's a couple of other things to know about cells. The fact that cells are open systems to both matter and energy. Remember, I'm talking about the cell, not the biosphere. I'm talking about the cell. The cell is open, is an open system in every way. That means that we are continuously running these reactions. The cell never reaches a state of equilibrium where no more reactions until it dies. The normal feature of life is that all of these reactions run continuously, and that is because we are continually taking in matter, losing matter, continually taking in energy and losing energy. That is what drives the phenomenon of life itself, is the continual need to be running these metabolic reactions. So the other thing to say here, it's no good just having a reaction which releases energy. That energy could all be in a form which is useless to the cell. Instead, this energy here must be, in a biological system, must be put into a form which is biologically useful. And we'll talk about the principal way in which that's done in a moment. So what is this? What we say the energy is the capacity to do work. What are the three kinds of work inside the cell that are most important? First of all, chemical, all of the chemical processes. Second of all, the work involved in transport and in moving stuff around inside the cell and moving the cell and everything else. The third is mechanical, is the mechanical work of building, but the mechanical work, for example, of gross movement and that kind of thing. So to do work, the cell's going to require all of these sources of energy in order to perform its work. We do that by coupling exergonic and endergonic reactions. And the way in which we couple them together is to take an exergonic set of reactions, take that free energy, and put it into a substance, usually the substance ATP. Most energy coupling in cells is done by the substance called ATP, adenosine triphosphate. And we'll see its structure in a minute. There are one or two instances where other substances can be used. But by and large, ATP is by far the most common. And if you understand how ATP works, you understand how the other more rare ones also work. So this is ATP. This is adenosine triphosphate. You think back and you'll realize when you look at this, it should look familiar because this is actually a nucleotide. Here it has a ribose sugar there. Here it has an adenine there. And here it has 3 phosphates there. This is a nucleotide, adenine, which could actually be used in RNA. It's a building block for nucleic acid, specifically for RNA because it uses a ribose sugar, not a deoxyribose. If this was deoxyribose, it could be used in DNA. This is a ribose sugar, so this is a nucleotide, and it is included in RNA when RNA is made. But inside the cell, adenosine with 3 phosphates attached to it is the commonest trafficker of energy. And the most of the energy that we are interested in is lodged in this bond here, in that bond between these 2 phosphates and the last phosphate. Under some circumstances, the second bond can also be used, but it's much more common just for this one to be used here. And the way in which this happens is that here is an adenosine triphosphate here. There is a hydrolysis reaction. In other words, we put in a water molecule. That bond there is severed, and inorganic phosphate results, but it produces energy. But it is very important to realize that this energy just doesn't like fly off the adenosine triphosphate. It is usually involved in transferring energy by creating a bond with this phosphate, by transforming this chemical bond here into another bond, often on a substrate or on an enzyme or something like that. Somehow or other, this phosphate with its attendant electrons is bonded, and that is how to another substance, and that is how the energy is actually transferred to whatever it is that we want to transfer it to. We don't simply, we don't simply release energy like in a light flash or whatever. It has to be, it goes in the form of energy, of electron transformations as well. So that reaction of ATP is actually highly exergonic. There is a lot of potential energy lodged in this bond here. We will learn later on how the cell actually does this, how the cell takes fuel, for instance, and transforms the energy contained in fuel into the potential energy lodged in this bond. That is a lengthy process that we will discuss. ATP is deliberately made, and it's deliberately made to contain the energy. The energy here originates from fuels that the cell transforms. So when we couple reactions like that, then we can actually get reactions to occur that would not happen spontaneously. What we're going to do, we're going to see that we're actually going to energize the substrates, destabilize them in such a way that their electrons now create new bonds that would not otherwise spontaneously have arisen. Here is an example. We have a great need in our cells for this. This is an amino acid called glutamine. And it is synthesized inside the cell. And it is synthesized by taking glutamic acid and ammonia. And the two are put together to form the amino acid glutamine. That reaction is a highly endergonic reaction. It requires energy to be put in for it to proceed. And that energy is, under normal circumstances, not freely available. It's not going to, that reaction is not going to proceed to equilibrium just spontaneously. Instead, what the cell does, it couples the breaking of that third phosphate bond on ATP. It couples that exergonic reaction to this reaction as follows. Here's glutamine. We react ATP with glutamine. In fact, it's done by an enzymatic system, but we don't have to worry about that for the moment. What we're watching is how this glutamine now is phosphorylated. It has a phosphate attached to it. It is phosphorylated. That phosphorus there has been extracted from the ATP. This is an exergonic reaction. And the energy is lodged here. Some, of course, is a little bit, don't forget, lost this heat. But we produce ADP, adenosine diphosphate. And here is the third phosphate, together with its highly energetic bond now attached to the glutamic acid. And this glutamic acid now is phosphorylated. In the process, what has happened is that, in fact, the glutamic acid now forms a phosphorylated compound, which is very unstable. All its electrons are actually discombobulated by this reaction. Some of the electrons are excited into unstable formations around their atoms. And when we actually now bring ammonia near to it, it is very easy for those destabilized atoms to react with the ammonia and form a new set of bonds. And here it is here. A new set of covalent bonds have been formed from those highly energized electrons, able now, they had sufficient kinetic energy in those destabilized bonds to form a new set of bonds. And that is our very stable product at the end of these reactions. Here it is glutamine. And when we look at the overall reaction, we take glutamic acid, ammonia, ATP, and we end up with glutamine, ADP, and this free phosphate group. So that is the way in which we're coupling. With this reaction, glutamic acid to ammonia is a highly endergonic reaction. It requires an input of energy. This reaction, the ATP, the phosphorylation of the dephosphorylation of ATP and the phosphorylation of the glutamic acid, that overall is exergonic, and it provides sufficient energy for this reaction to occur, to form these new covalent bonds between glutamine and ammonia. That is how we couple. We'll see that kind of coupling all through biological systems. The other thing, just to emphasize, many of these reactions, we're going to find out, many of these reactions are shepherded by enzymes. They are what ensures that the reactions take place at a fast enough rate to be biologically useful. Okay, so here are some real instances, we saw that we talked about some of the work that needs to be done inside the cell. Well, let's have a look specifically at the proteins. Proteins very frequently interact with ATP, and it is the interaction, the phosphorylation, dephosphorylation, phosphorylation, dephosphorylation of proteins is often what drives the work that they do. And here is a good example. This is an example of active transport at the membrane, and this is a transport protein. This transport is against the concentration gradient. This transport protein is moving a substance, a solute, whatever, to an area of already high concentration. And in order to do that, that is not something which is going to happen spontaneously. That is an endergonic reaction. And we're going to have to obtain energy from somewhere in order to drive that. And this is how it's done. The protein itself interacts with ATP and a phosphate and becomes phosphorylated. That phosphorylation induces a structural change in the protein. It causes the protein to change shape. And the protein, in this case, it's shown simply, it kind of inverts, and so that the opening is facing the other way. When that happens, the protein loses its affinity for that solute, and the solute leaves to enter the solution here. And the protein is dephosphorylated in that process of release and returns to this shape here, ready to be phosphorylated again, to do another pump. So it phosphorylates, pumps, dephosphorylates, and returns to its original shape. All of that requires energy. And the energy is coming from that phosphate bond there. That is what the energy contained in that phosphate bond is you can think of it in a way as it's destabilizing the structures of the protein and causing it to change shape. And once it's changed shape and it dephosphorylates, it returns to its old shape. We have converted the potential energy of that phosphate bond into the actual energy of movement and of transport, that the movement of the protein ends up transporting the substance. Here's another one where this is actual real movement. And this is the source of a lot of our movement. This is the action of those little motor proteins that we talked about when we talked about the cytoskeleton. A lot of motion in the cell, and in fact, gross motion, such as our muscle movement, arises because of an interaction between motor proteins, shown here with their two little feet, and some cytoskeletal element, very often actin or a microtubule in the cytoskeleton. So here, this is how this works. This motor protein has a kind of a native shape. And when we phosphorylate the protein, we induce a shape change in the motor protein. In this case, the motor protein swings one leg forward and fixes. And when that happens, well, as soon as it's fixed, it dephosphorylates and it returns the other leg around to its original shape. So each time you phosphorylate, step, dephosphorylate, and go back to your original shape. Phosphorylate, step, dephosphorylate, return to original shape. And that is how the motor protein works as it moves along the track, either of a microtubule or of an actin filament. The gross movement of muscle, the contraction of muscle, works in a similar way, excepting that the motor proteins interact with actin, but their motor proteins are fixed in place. So the motor proteins, they undergo a similar structural change. They'll reach out, grab actin filaments, and pull them and dephosphorylate and release. That's how actin filaments in muscle are pulled towards one another. It's the same idea. These motor proteins, when they are phosphorylated, undergo a structural change in shape. Once that change is complete, they are dephosphorylated and they return to their original shape. Okay, so ATP is constantly required in the cell, but I should tell you that ATP is very, very short-lived. This is not an energy store. It's a means of rapid energy transformation. We're rapidly converting from one form, often chemical energy, potential energy locked in chemical bonds of fuel, say, into a very temporary, useful biological form, which is immediately contributed to a reaction of some sort. But we have to renew our ATP the whole time. We have to take ADP and we have to continually phosphorylate it to contribute to those reactions. And that the energy contained in that bond is derived from fuels during catabolic reactions. So there is this continual generation of ATP, and then it's dephosphorylation to ADP, then it's regeneration to ATP. It's a constant feature of the metabolism of the cell. And that transformation links the catabolic and the anabolic reactions of the cell. As here, we have ADP and free phosphates here. That reaction there does not proceed spontaneously. We require input of energy from catabolic reactions. And we're going to put that energy in here and create a bond there. I should tell you that bond is highly energetic and very unstable. That's why ATP is rather short-lived. But the other thing is this here, it's a simple sentence. Energy from catabolism, from exergonic energy releasing processes is put in to make the ATP. But this here is a very complex process. And we will talk about it at length in another class when we talk about respiration. When we talk about cellular respiration, that is what this process is. Cellular respiration is the process whereby we take energy from fuels and we convert it into biologically useful energy in the form of ATP. That ATP, once it's made, the energy lodged in that phosphate bond here can be contributed to otherwise endergonic reactions, and that provision of energy will allow them to proceed where they would not otherwise. Okay, and so we couple energy from catabolism to anabolic reactions via this carrier molecule ATP. Okay, so having said all of that, we also need to realize that most of our biological reactions in the cell, while we can certainly see how they occur, most reactions, in fact, would proceed very, very slowly, if at all, if it were not for the presence in the cell of enzymes. Now, an enzyme is a catalyst. An enzyme is a substance which is unaffected by a reaction, but participates in the reaction in order to facilitate it. It's a catalyst. Here's an example of what we mean. This is a substance, sucrose, our common table salt. It consists of a glucose molecule and a fructose molecule joined by a very strong covalent bond. So the thing to think about is this. If I was to take sucrose, table sugar, and I just simply leave it in a bowl, the question I need to ask you is this. Does that sucrose ever break down spontaneously? Does this ever break down spontaneously? Well, in order to break down, this molecule here has to be destabilized somehow. We have to destabilize this bond. There are various ways that can happen. A cosmic ray can hit it, right? All molecules have inherent kinetic motion as long as they're above absolute zero. So the molecules are also vibrating around. Well, you could have a whole lot of other molecules just randomly smack into this one and contribute kinetic energy, which destabilizes that and break it. So the answer is yes. If I leave table sugar alone, some of the molecules will break down every now and again. They'll get a little burst of energy from some source, and they'll be able to break. But that will happen extremely slowly. Those reactions would be very, very rare. You could come back in thousands of years' time and find that hardly any of your table sugar had broken and formed a glucose molecule and a fructose molecule. But the cell very frequently needs to use sucrose, and it needs to break sucrose into its component monosaccharides, probably to use them for fuel. It could be using them for some other things if it's a plant, but usually it would be breaking them to start using them as fuel. The cell can't wait around for this to happen spontaneously. Instead, the cell has to have some way of managing that reaction and ensuring that it happens when it needs to happen and that it happens efficiently. And to do that, it makes use of an enzyme. In this case, it is the enzyme sucrase. Whenever you see that ASE on the end of a word, you know that that is an enzyme. This is sucrase, the enzyme, which breaks down sucrose. Sucrase is a protein. 99% of the enzymes in our bodies, in our cells rather, are proteins. There's one exception, and that is RNA. RNA, under certain circumstances, certain species of RNA can act as enzymes, as we'll hear later on. They are referred to as the ribozymes. But most of our enzyme activity is by proteins, and the recipe for those proteins is lodged in the genes. Most of DNA is there to record the recipe for our enzymes, and our enzymes drive the metabolism of our cells by speeding up reactions that would otherwise happen so slowly they'd be useless to the cell. So that is the primary function of the enzymes. And here it is. Here you can see that it actually induces the hydrolysis of this molecule and allows glucose to be freed and fructose to be freed. Those probably then go off to other biological reactions to be used. So that is the role of sucrase here. And we got to ask the question, what is the sucrase actually doing? How does it achieve that? Well, we need to think about how these reactions occur anyway. I said to you that if we just leave the table sugar, it's going to require a small input of energy to get anything to happen at all. It's not just going to spontaneously break unless we heat it up so much that its own kinetic energy gets so great that it destabilizes the bond. We've added energy in the form of heat. Or it gets smacked by other molecules, which destabilize, but it's also an input of energy. That little input of energy to get the reaction to start happening is called the energy of activation. And here it is here. Here, we're going to consider here two sets of reactants. This compound here, AB, joined with a covalent bond, and compound CD, joined with a covalent bond. We can mix them. Maybe every now and again, a pair of them will get enough energy to actually smack together and be destabilized. And when that happens, they enter a state where they are in a transition. Their bonds are so destabilized that the bonds can reform. And as they start to reform, they form the new compounds AC, BD. If we were to look at the free energy, we'll find we actually had to put in that little bit of energy there. And once we've pushed them into the transition state, then the reaction occurs spontaneously to produce the products with the release of energy here. So this looks to us like the barrier. The amount of energy you have to put in at the start of that is a barrier to the reaction proceeding spontaneously. And that is called the energy of activation. And for many biological reactions, that energy is extremely high. If we are wanting to build large biological molecules, for example, the energy of activation is large, and it is very difficult. Those reactions will only occur extremely slowly. There's only every now and again you'll get them to spontaneously rearrange their bonds, spontaneously acquire enough energy to rearrange the bonds. You have to actually put in a large amount of energy if you want them to proceed. And the energy of activation acts as a barrier. This is what enzymes do. Enzymes catalyze a reaction because they lower this barrier. They lower the energy of activation. They don't affect the overall dynamics of the reaction. That is all that they do. They lower the energy of activation required. They do it by various means that we'll talk about in a minute. But their net effect is that they hasten reactions that would otherwise occur so slowly they would be biologically useless. So here's a reaction without any enzyme. Here is the reaction with enzyme. And the energy of activation here is much lower. If we were to look at the dynamics of the reaction, we would find that the delta G, the free energy change, is not affected. But this energy of activation is a tremendous barrier to the initial reactions that would start the reaction to allow it to go to completion. That is what the enzymes do. They lower this barrier. And the result is that enzymes they never enable an impossible reaction to occur. They do not make impossible reactions possible. They take reactions which are eminently possible, but which happen very, very slowly, and they speed it up. And that does two things. First of all, it makes it efficient for the cell. It means that the reactions happen at a rate which is useful to the cell. But the second thing is it gives the cell control. If the enzyme is not present, the reaction's not going to take place. If the enzyme is present and active, then the reaction starts. We may only want a reaction to take place for a certain length of time. If we want to do that, we can enzymatically control whether or not that reaction happens. So we now need to ask the question of how is it that enzymes actually do it? Well, the first thing is the reactants that enzymes react with. The enzyme is extremely specific for its substrates, very, very specific. It will only react with particular substances, and that is because a lot of the action of the enzyme depends upon its shape, and it has to fit the substrates perfectly. That is why you have an enzyme, it has substrates. If you change the substrate even slightly, the enzyme can no longer bind to it. When the enzyme binds to its substrate, which is the start of this reaction process, it forms a complex between the enzyme and the called the enzyme substrate complex. And once it's done that, then we'll see that the enzyme can do various things to the substrate in order to speed up their reactions to one another, or the reactions. It's not necessarily to one another, it's to speed up the reactions. That place where the substrates bind is called the active site. And the substrate fits in there by a process of induction. It's called the induced fit, because it's not only the shape that the shape of the enzyme active site must match the shape of the substrate, but it also matches chemically. You would have, for example, you would have charged groups on the substrate are attracted to charged groups on the enzyme, that sort of thing. So it's not only the shape, but also it's chemical makeup that allows that the substrate to fit in and to fit tightly and properly into the active site. And that close fit actually can induce changes in the shape and structure of the enzyme itself, which may contribute to the catalysis. So have a look here, and you'll see what I mean. Here's the active site for this particular enzyme, and here is its substrate. You'll notice something. The enzyme may be absolutely enormous compared to the size of the active site. This is not useless. All of these amino acids that constitute this very complex polypeptide that is formed into this protein enzyme, all of these things will interact in particular ways once we allow the substrate to bind here. For example, in this case here, the substrate is bound and the whole shape of the enzyme has changed. It has moved, can you see? It's like closed up around the substrate. And there are things happening. New chemical groups, perhaps on amino acids here, are being pressed into the substrate. Bonds in the substrate are being stressed and strained now by the change in shape of the protein. All sorts of things are actually happening, actively happening in this enzyme substrate complex. And they are going to induce changes in the substrate. And that, for example, let's think that this is sucrose, right? Sucrose, the glucose molecule and a fructose molecule. Now we brought it into the sucrase. When we bring it into the sucrase changes shape and starts to stretch those bonds between the glucose and the fructose until they break. Once that reaction has taken place, the substrate no longer fits. It no longer fits the active site. And this enzyme substrate complex, now the enzyme actually will revert to its old shape. It no longer has any attraction for the products that have resulted, glucose and fructose. Its substrate is sucrose, not glucose and fructose. And it will release the glucose and fructose, and it will sit there ready to receive another substrate molecule and do the same thing again. So this binding into the enzyme substrate complex, it can, at the active site, is what results in the lowering of the energy of activation, because the enzyme, it can be orienting substrates in a particular way so that a reaction will happen easily. It can be straining substrate bonds, the way I described for sucrose, where it actually stressed the glucose molecule and the fructose molecule till they broke. Sometimes the enzyme will provide a favorable microenvironment, and it also can induce covalent bonding to the substrate of various substances. I should also say that enzymes are sometimes phosphorylated by the breakdown of ATP, and can contribute energy to new covalent bonds in the products relative to the substrate. So there are all sorts of things that the enzyme actually can do once that enzyme substrate complex has formed. And all of them contribute to lowering the energy of activation and enabling a reaction to take place rapidly, that would otherwise take place very slowly. So here we see, we have substrates here. Those substrates bind to the enzyme, forming an enzyme substrate complex. And they are held in place very often by weak interactions such as hydrogen bonding, hydrophobic bonding, van der Waals forces, those sorts of weak bonds, weak interactions rather. The presence of the substrates in the enzyme induces some change in the enzyme. It causes the enzyme to do something and to interact with the substrates. And in doing so, this reaction between the substrates is enabled. The energy of activation is lowered so that these substrates here react with one another in a specific way to produce a product. The product does not have the same attraction to the enzyme as the substrate. The product actually is released from the enzyme because it has changed. It is no longer, there is no longer an induced fit to the active site, and it is released. And now the active site, the shape of the enzyme changes back to its original, and the active site now is available to do the same thing over again. This is another characteristic of the enzymes. They will operate over and over and over and over and over again. They repeat the same reaction very, very rapidly. And they can do the reaction many thousands of times. As a result, we only need a small amount of enzyme very often to perform a lot of work, to run through a lot of these reactions very, very fast. So the enzymes that we're going to be talking about for the most part are proteins. And the shape, you'll remember thinking back, we talked about how proteins are actually structured. First of all, there are a chain of amino acids joined together. That's the primary structure. Then those amino acids, they have regularly repeating groups associated with them, and those groups interact with one another to form either helices or sheets, alpha helices, beta plated sheets, or sometimes there's a little area with no structure which acts as a hinge where it can be bent in space. then those large areas of that polypeptide interact with one another to start folding the polypeptide into a specific shape. And then finally, sometimes different polypeptides will become associated with one another to form the mature protein. So those are the 4 levels of protein organization. But you'll realize that a lot of those interactions that produce all of that structuring in 3-dimensional space, a lot of those interactions, for example, are through hydrogen bonding, through interactions of charge, and these sorts of things. Those are some of the structuring is through strong covalent bonds, particularly across sulfur groups. But much of that 3-dimensional folding and structuring in space is through things like hydrogen bonding, which are weak. And hydrogen bonding, those kinds of bonds, are very much affected by environmental conditions, especially the pH. So that's the first thing we'll think about is, if I change the pH, what am I doing? I'm changing the relative proportion of protons, H+, or hydroxyl ions, O-. Can be radically affecting the overall charge of the contained in the environment, and be affecting the structure of the hydrogen bonds. So it's no surprise then that the pH affects the protein structure, and in an enzyme affects its function very much. The second thing that radically affects enzymes and protein structure in general is temperature. And we'll find out that, in fact, most enzymes have an optimum temperature at which they operate, and an optimum pH at which they operate. So let's have a look here and see, first of all, temperature. Here is an enzyme which is operating at around about 37 degrees is its optimal. That's where it works best. That's where it works most efficiently. It will continue to work at lower temperatures, but its efficiency falls until it reaches a minimum temperature, at which point activity stops. Similarly, it has a maximum temperature. And you'll notice something. Normally in chemical reactions, when we increase the temperature, we increase the rate of reaction. But enzymes are different. They are living things. And they have a maximum, they have an optimal temperature at which they operate. If we go above the optimal, for a little while, they will continue to work, but they work less and less efficiently until activity stops. Now, there's one other thing to say, and that is this minimum temperature here. For most enzymes, if I now increase the temperature, they will begin working again. Dropping them to low temperatures, there are some which are fussy, but most enzymes, you can actually cool them way down and then warm them, and their activity would be restored. That is not true for raising the temperature. Very often, for most enzymes, if you raise the temperature far above their optimum, when they reach this point here, they are dead. The enzyme is irrevocably damaged. It is denatured by high temperature, and its activity cannot be restored. So this is very important when you think about environmentally, especially for organisms which can have no control over body temperature, which are dependent on environmental temperature. Their maximum and minimum temperatures are critically important to their function. For us, we maintain our body temperature here at 37 degrees, and all of our enzymes are basically, they are adapted to that particular degree. They do not function well above or below our optimum. Enzymes, most of them are proteins, and they are very subject to high temperature, to being denatured by high temperature. Just think about egg white. Egg white's not an enzyme, but it's a protein. And just think about what happens to egg white. That's an example for you of the radical changes that raising the temperature too high can have on the structure of a protein. Have a look here And you'll see here is an enzyme which operates, its optimum is right up here at about 77, 76, somewhere around about there. And here's its maximum is right up at 90. Has a minimum, the minimum is 40 degrees, which is already really, really warm, really hot. And for our enzymes would be a very dangerous temperature. But this enzyme here is adapted to these high temperatures. And this is typical of organisms, for example, which live in very hot environments, such as hot pools or hot springs. And these are especially prokaryotes, bacteria, that are specially adapted to that kind of environment and which do not function well at lower temperatures. Their proteins and, by the way, their nucleic acids are adapted so that they operate at a high temperature. There's a similar idea for pH. Enzymes have an optimum pH at which they operate, and a maximum and a minimum pH above and below which they will no longer operate. Away from their optimum, their efficiency is reduced. Here are two different enzymes. This one is adapted to operate at the pH of 2. It's very, very acid. This one is adapted to operate at about 8, pH of 8. Well, we in our bodies have enzymes whose optima lie right here. This one here would be typical, for example, of the enzyme pepsin, which is produced in the stomach. And the stomach is kept at a low pH like this, specifically so that pepsin is active. And it has a very narrow pH range. And so that it requires a low pH in order to operate. So this one here is a pancreatic enzyme, and this is trypsin. This one breaks down proteins. This one also breaks down proteins. But this one is trypsin released by the pancreas, and look, its optimum pH is 8. So now you have to think, well, the stomach and the pancreas lie close to one another, and they interact through the intestine. The stomach releases its contents into the small intestine. It moves down the small intestine, and it reaches a point where it encounters ducts from the pancreas. The way in which this works is the acid contents of the stomach are released into the intestine, but the pancreas releases bicarbonate, which is alkali, and it raises the pH to 8. The pepsin rather stops working. It no longer works because it's way outside of its pH range. But now the environment now is alkali, and now the trypsin, also released by the pancreas, becomes active, and its optimum is at around 8. For most of our cellular reactions, occur around neutrality, around 7. A little bit high, a little bit low, sometimes depending on particular organelles, but most cellular reactions occur around 7, around neutrality. Oh, sorry, it's the same things. Okay, so those are the basic ways in which enzymes start to operate. Enzymes do have some other things associated with them. They're often not just proteins. They're often very complex compounds of different substances. And one of the most important are cofactors. These are substances. Very, very often, these are inorganic substances associated with the enzyme. And for example, there are metals which are associated with some enzymes. The one example which you will know of is the enzyme hemoglobin. Hemoglobin is a protein. It is an enzyme. and it picks up oxygen in the lungs, transports it to areas with low oxygen, and releases the oxygen. And the hemoglobin has a cofactor associated with it, which is an iron atom that sits right in the middle and forms a group called the heme group, which donates its name, hemoglobin, to the whole thing. But there are many of these, many enzymes which have these cofactors associated with them. They may also have an organic cofactor. And organic cofactors are called coenzymes, and many of our vitamins are in fact coenzymes. So that is one of the reasons for the importance of vitamins. They interact, they form part of an enzyme complex, which enable the enzyme to operate efficiently. Okay, so I tell you what I want you to do. I want you to just stop here. Just for 5, I will continue in 5 minutes time, but I want you just to have a break, okay, for 5 minutes. And I just want you to go through the slides that we've covered so far. We've covered an enormous amount of material. And I just want you to take 5 minutes, clear your heads, flip through what we've done, and then we'll return. I've just got a short thing that I want to say after this, and then we'll be done. So just take 5 minutes, please, clear your mind. The reason I want you to do that is because the next bit is an important section. 5 minutes. Okay, you're back. We said that enzymes are one of the principal ways in which a cell controls its metabolism. The first thing we realize, the control is exerted because enzymes enable reactions to take place at a rate which is biologically useful. Otherwise, those reactions would take place so slowly they would not be useful. So that is the first thing. If the enzyme is present at all, then it will facilitate the reaction. If it's not present, it's not going to facilitate the reaction. So that gives us one way of controlling the enzyme. If an enzyme is not needed continuously, some enzymes we have to have operating continuously in the cell. Others we only require every now and again. If we only require them every now and again, one control we can exert is to only read off the genes that produce them when they are required and stop reading off the gene when they are not required. But there are other ways to control enzyme activity as well. Now, the first way is an external control. This is not a system which is used by the cell to control its own enzymes. This is a system used by other organisms to control another organism's enzymes, for example. You'll understand what I mean in a second. We're going to talk about the use of inhibitors, which actually stop an enzyme from working. And the first type of inhibitor we'll talk about are the competitive inhibitors. And a competitive inhibitor is one which binds to the active site of an enzyme, and it binds permanently to that active site. It has just enough similarity to part of the active site that it can occupy it. And in doing so, it actually causes the enzyme to stop working because a natural substrate cannot get in. It cannot get in to operate. This is a very dangerous situation because what you've in fact done is you've killed the enzyme activity. And this is competitive inhibitors are typically toxins and poisons. They are things which come in from outside, from something else, which induce, and there are many of these that we can think of. For example, snake venoms are often competitive inhibitors for various enzymes in the body. Mushroom toxins, there's some very, very, very fierce mushroom toxins which are competitive inhibitors. And a simple example which for you is an actual inorganic compound, such as hydrogen cyanide. Cyanide is a compound, a relatively simple substance, CHN, which will fit in to an enzyme, one of the enzymes in our respiratory pathway, one of the last enzymes in our respiratory pathway, has an active site which can be blocked by a cyanide. And that is why cyanide is such a powerful poison. If somebody is poisoned with cyanide, their respiratory chains all stop. And in fact, their cells suffocate, but you suffocate at a cellular level because you simply can no longer do the processing needed to make ATP. That's what it does. So competitive inhibition is a system where something outside controls enzyme activity. It is not a way in which the cell controls its own enzyme activity. But there is a very subtle way in which the cell can control its own activity. And this is through a system which we call non-competitive inhibition. There are various ways in which this takes place, but here is a very simple example. Here is an enzyme, and here is its active site. Okay, this is its normal substrate. This is where it will normally bind the substrate. But elsewhere on the enzyme, there is another site here, and that is called the allosteric site. And the allosteric site can bind something which causes the shape of the enzyme to change. And the active site is no longer active. So here it is here. This is called an allosteric inhibitor. It is binding remote from the active site, changing the shape, in this case, of the active site so that it no longer can perform the catalysis. This is reversible. It's completely reversible. If I take this off, the enzyme is restored to its active form. This gives the cell a very subtle way of controlling its own enzymes, as we'll see. And in fact, it also gives us a way, we'll see, of activating. So this is a way in which the cell can have an on and an off switch, is by allosteric interactions. So I think you know that I'm actually going to stop there. I think that we've actually had enough for today, and we'll carry on with this next time. I just have the feeling that maybe this is better left for when your brains are fresh. Okay, so I'll see you then next time.